Hello friends, today I am here with second part of photosynthesis in higher plant of 11th standard. In this session, we will discuss about phosphorylation and photophosphorylation, cyclic photophosphorylation and non-cyclic photophosphorylation, chemiosmotic hypothesis, utilization of ATP and NADPH, primary acceptor of carbon dioxide and Calvin cycle. First, we will discuss about phosphorylation and photophosphorylation. The process through which ATP is synthesized by cells in mitochondria and chloroplast is called phosphorylation. And photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of sunlight. So this is the difference. In case of phosphorylation, no sunlight is used, but in case of photophosphorylation, the synthesis of ATP is done in presence of sunlight. Now it is about cyclic photophosphorylation. Cyclic photophosphorylation involves only photosystem 1, that is PS1, and it is not concerned with the photooxidation of water. The electrons which is excited from photosystem 1 is again returned back to photosystem 1. Here there is production of ATP. It occurs only under special conditions. And what is the special conditions? When NADPH starts accumulating. Here in the given figure, you may observe the cyclic photophosphorylation. The excited electrons is cycled back to photosystem 1 complex through the electron transport system. Now let us discuss about non-cyclic photophosphorylation. Non-cyclic photophosphorylation involves both photosystem 2 and photosystem 1. In this case, the first step is photooxidation of water, resulting in the splitting of water into hydrogen, electron and release of oxygen. Here there is sequence of electron transfers where NADP plus is reduced by photosystem 1. Then photosystem 1 is reduced by photosystem 2 and further photosystem 2 is reduced by water. Non-cyclic photophosphorylation is also concerned with the reduction of NADP plus and production of ATP, and this takes place under normal condition. Now it is about chemiosmotic hypothesis. According to chemiosmotic hypothesis, it is discussed about the mechanism of synthesis of ATP in chloroplast. Like respiration, in photosynthesis also ATP synthesis takes place, which is linked to the development of a proton gradient across a membrane of thylakoid. So let us understand this in 3 steps. In the first step, since the splitting of water molecule takes place on the inner side of the membrane, the protons or hydrogen ions are produced by the splitting of water. These hydrogen ions get accumulated within the lumen of thylakoid. In the second step, as electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. In the third step, since the NADP reductase enzyme is located on the stroma side of the membrane. So along with electrons that come from the acceptor of electrons of photosystem 1, protons are necessary for the reduction of NADP to NADPH and H+. These protons are removed from the stroma. Now, so let us see how this happens. Due to difference in hydrogen ion concentration on the two sides of the thylakoid membranes leads to the building of a transmembrane proton concentration gradient. Further, this electrochemical potential gradients of proton, that is high hydrogen ion inside the thylakoid and low hydrogen ion outside in stroma, acts as the driving force behind the conversion of ADP and inorganic phosphate into ATP. The formation of ATP is catalyzed by enzyme ATPase, which is located in the thylakoid membrane, but the thylakoid membrane is not permeable to protons. They move towards stroma side of the membrane through ATPase enzyme complex, which consists of two parts, that is coupling factor 0 and coupling factor 1, denoted by CF0 and CF1. The coupling factor 0 is embedded in the membrane and forms a transmembrane channel to facilitate diffusion of protons across the thylakoid membrane. The coupling factor 1 is present outer stroma side of the membrane where ATP is synthesized. So it is now well established that one molecule of ATP is formed for every 3 hydrogen ion passing through ATPase complex. The given figure clearly shows about ATP synthesis through chemiosmosis. You may also observe here the location of coupling factor 0 and location of coupling factor 1. Now the next topic is where are ATP and NADPH used? As we know that the products of light reaction are ATP and NADPH and also oxygen. Oxygen diffuses out from the chloroplast into the atmosphere, whereas ATP and NADPH are used in the synthesis of food, particularly sugar. So this is called biosynthetic phase, which does not directly dependent on the presence of light, but it is dependent on the products of light reaction, that is ATP, NADPH besides carbon dioxide and water. Now let us see where is the which is the primary acceptor of carbon dioxide in dark reaction. So the primary acceptor of carbon dioxide in dark reaction is 5-carbon ketose sugar, that is ribulose biphosphate, RUBP. In the given reaction below, you may observe the carbon dioxide is accepted by ribulose 1,5-bisphosphate to form 2 molecules of 3-phosphoglycerate. Now we will discuss in detail about dark reaction, that is Calvin cycle. Calvin cycle is described under 3 headings, that is carboxylation, reduction and regeneration. So let us discuss here one by one. First we will discuss about carboxylation. Carboxylation means fixation of carbon dioxide into a stable organic intermediate. Here CO2 is utilized for carboxylation of ribulose bisphosphate in the presence of enzyme ribulose bisphosphate carboxylase, or better it is ribulose bisphosphate carboxylase oxygenase, or we call it as Rubisco. Due to this, two molecules of 3-phosphoglycerate is formed. Now the second step of Calvin cycle is reduction. During reduction process, there is formation of glucose. It involves utilization of two molecules of ATP for phosphorylation and two molecules of NADPH for reduction per CO2 molecules fixed. The fixation of 6 molecules of carbon dioxide and 6 turns of cycle are required for the removal of 1 molecule of glucose from the pathway. Third step is regeneration. The regeneration steps require 1 ATP for phosphorylation to form ribulose bisphosphate. Hence, for every carbon dioxide molecules entering in the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. Since there is 6 molecules of carbon dioxide and 6 turns of cycle, so altogether 18 ATP and 12 NADPH are required. It is shown in the given table. In the given figure, you may observe the 3 steps of Calvin cycle. In carboxylation, carbon dioxide is combined with ribulose 1,5-bisphosphate. In reduction, carbohydrate is formed at the expense of the photochemically mode of ATP and NADPH. In regeneration, carbon dioxide acceptor ribulose 1,5-bisphosphate is formed again, so that the cycle continued. So this is all about today's session. Thank you. For any kind of doubts, please write your question in comment section.